Good day to you, sir. So this is my presentation of the midterm lecture exam. The first question is the parts of my flower and how it develops into a fruit. So this is the flower that I have found outside of my house. It has a petal, the stigma, the style, the ovary, and the ovule, which makes up the pistil, and below is the sepal and the stem. So how it develops into a fruit. The first step of fruit formation starts with a pollinated flower. Some flowers pollinate themselves, while others depend on wind, humans, animals, insects, or other methods to bring pollen and ovules from different or the same plants together. For species that depend on cross-pollination, weather conditions and the number of pollinators can be a factor in how many flowers actually become a fruit. The female part of the flower is referred to as the carpel, and it consists of a long slender tube called the style, which leads down to the rounded base, which is the ovule-filled ovary. Surrounding the ovule is the pericarp, which matures into what we consider the fruit. On top of the style is the stigma, which is generally sticky in order to catch pollen. The male part of the flower is referred to as the stamen, and it consists of a long slender filament topped by the pollen-containing anther. When a grain of pollen reaches the stigma, it creates a pollen tube for the sperm to journey down the style and fertilize the ovule. Fertilized ovules become seeds. Fertilization is the death of the flower, as the petals drop or wither at this point, and the ovary starts to enlarge and ripen into what we know as the fruit. After pollination, there are many things that can happen while fruit is forming. Weather conditions greatly affect yields. Fruit needs a lot of water to develop, and if there's not adequate rainfall, fruit may fail to grow if not irrigated properly. Temperatures will also affect fruit set and ripening, as will hail and strong winds. Pests and disease are other problems that can decimate a fruit crop too. So this is an illustration of the process of how a flower becomes a fruit. So first, the flower just after fertilization, and the embryo develops, the seed develops from ovule, fruit develops from ovary, and fruit ripens, flower parts fall away. So this is the second question, the difference of photosynthesis and respiration. So what is the difference of photosynthesis and respiration? The relationship between photosynthesis and cellular respiration is such that the products of one system are the reactants to the other. Photosynthesis involves the use of energy from sunlight, water, and carbon dioxide to produce glucose and oxygen, while cellular respiration uses glucose and oxygen to produce carbon dioxide and water. To emphasize this point even more, the equation for photosynthesis is the opposite of cellular respiration. Humans, animals, and plants depend on the cycle of cellular respiration and photosynthesis for survival. The oxygen produced by plants during photosynthesis is what humans and animals inhale for the blood to transport to the cells for respiration. The carbon dioxide produced during respiration is released from the body and absorbed by the plants to help provide the energy they need for growth and development. The process of photosynthesis is used by plants and other photosynthetic organisms to produce energy, whereas the process of cellular respiration breaks down the energy for use. Despite the differences between these two processes, there are some similarities. For example, both processes synthesize and use ATP, the energy currency. The cycle of photosynthesis and respiration maintains the balance of oxygen and carbon dioxide. Photosynthesis produces the oxygen to replenish oxygen that is used up by the living organisms during respiration. Carbon dioxide produced during respiration is one of the reactants plants need to perform photosynthesis. Photosynthesis and cellular respiration are both part of a mutually beneficial relationship. Cellular respiration cannot occur without photosynthesis, and photosynthesis certainly cannot occur without the help of its partner. This is the never-ending cycle that sustains life on Earth. So the main difference between the photosynthesis and cellular respiration is that photosynthesis is an anabolic process, where the synthesis of organic compounds occur, storing energy, whereas cellular respiration is a catabolic process, where the stored organic compounds are utilized, producing energy. So this is an illustration of photosynthesis and cell respiration of plants and humans, respectively. So the next question is the process of fermentation, which I chose are the wines. So the process of fermentation in wines. At its simplest, fermentation is often described as the conversion of one molecule of glucose into two molecules each of ethanol or ethyl alcohol and carbon dioxide. While that's arguably the most important result, yeasts are complex organisms that perform a wide array of biochemical processes in a fermenting wine. The fermentation in wines, which we will discuss about esters, tannins, acetaldehyde, anthocyanins, sulfites, and amino acids, compounds found in wine fermentation. First are esters. Esters are aromatic compounds that contribute delicate fruity, citrusy, or floral aromas to a young wine. They exist in must as precursors that are bound to sugar molecules. As the yeast consume the sugar, the esters are liberated and become volatile. So this is the ester compound. Next are tannins. Present naturally in grape skins and seeds, tannins are antioxidant polyphenols that give wines dryness, astringency, and mouthfeel. The alcohol produced during fermentation enhances tannin extraction, while fermentation byproducts react with tannins, altering their structure and in turn their perceived levels of astringency and bitterness. So this is the tannin compound, very complex. Next are acetaldehyde. Created by yeast in the penultimate step of the pathway to ethanol, low levels of acetaldehyde can enhance fruity aromas in wine. In high concentrations, acetaldehyde may yield unwanted bruised apple-like aromas and flavors. Acetaldehyde abilities to catalyze tannin polymerization plays an instrumental role in the stabilization of red wine structure and mouthfeel. So this is the acetaldehyde compound. Next are anthocyanins. It is a highly reactive family of compounds in red grape skins, gives red wine its color and antioxidative properties. These compounds polymerize in the presence of acetaldehyde to form a vast array of stable color components. So this is the anthocyanin compound. Next are sulfites. As with ethanol, yeast produces sulfites during fermentation to fend off competition from other microorganisms. These natural sulfites can somewhat protect the wine from microbial spoilage and premature oxidation, but their levels are typically bolstered by winemakers after fermentation. And lastly are amino acids. Unfermented grape juice or must is rich in nitrogen-containing amino acids. Yeast consume most of these amino acids during fermentation, using the nitrogen to construct proteins and amino acids necessary to live and reproduce. Amino acids are the most important family of compounds in yeast nutrition and health. And this is the compound of amino acids. Over here, we'll talk about the process of fermentation. The process of fermentation in wines. First, glycolysis is the metabolic pathway that converts glucose, a type of sugar, into pyruvate, which is the first major step of fermentation or respiration in cells. Glycolysis occurs not only in microorganisms but in every living cell. In glycolysis, a single molecule of glucose. Zymase is a complex of several enzymes, and cozymase is a mixture of ATP, ADP, or adenosine diphosphate, a hydrolyzed form of ATP, metals, and coenzymes, substances that combine with proteins to make them functional, such as NAD+, or nicotinamide adenine dinucleotide. Both components were required for fermentation to occur. Next, in glycolysis, the two molecules of ATP are produced for each broken molecule of glucose. During glycolysis, two reduction-oxidation or redox reactions occur. In a redox reaction, one molecule is oxidized by losing electrons, while the other molecule is reduced by gaining those electrons. Next, a molecule called NADH acts as the electron carrier in glycolysis, and this molecule must be reconstituted to ensure continuity of the glycolysis pathway. Glucose is converted into pyruvic acid during glycolysis. Next, when oxygen is available, pyruvic acid enters a series of chemical reactions, known as the tricarboxylic acid cycle, and proceeds to the respiratory chain. As a result of respiration, cells produce 36 to 38 molecules of ATP for each molecule of glucose oxidized. In the absence of oxygen or anoxygenic conditions, pyruvic acid can allow two different routes depending on the type of cell. It can be now converted into ethanol, or alcohol, and carbon dioxide through the alcoholic fermentation pathway, or it can be converted into lactate through the lactic acid fermentation pathway. Several types of microorganisms, including yeast and some bacteria, have been used to break down pyruvic acid to produce ethanol in beer brewing and wine making. The other byproduct of fermentation, carbon dioxide, is used in bread making and the production of carbonated beverages. Other living organisms, such as humans, metabolize pyruvic acid into lactate because they lack the enzymes needed for alcohol production, and in mammals, lactate is recycled into glucose by the liver. So the last question is the difference between diffusion, osmosis, and active transport, and why these processes are important for plants. Diffusion. Diffusion is the movement of dissolved solutes or gases from an area of high concentration to an area of low concentration, down a concentration gradient. This is a passive process and so requires no energy in order to take place. It takes place because of random movement of the particles due to their inherent kinetic energy. In living organisms, diffusion of dissolved substances such as oxygen, carbon dioxide, and glucose occurs into and out of cells across the semi-permeable cell membrane. Semi-permeable means that only certain substances can cross the membrane. One example of this process are our lungs. In the lungs, oxygen passes from the alveoli into the bloodstream, and carbon dioxide goes the other way. Lungs are perfectly adapted for this exchange. So this is the process of diffusion found inside our lungs. Osmosis. Osmosis, on the other hand, is a special case of diffusion. It is the movement of water down a concentration gradient from high to low concentration across a partially impermeable membrane. In plants, osmosis is important to support the stems and leaves, and is through this process that water enters the plant cells. Water is stored in the cell vacuole, causing it to swell and press the cytoplasm against the cell walls. Plants need the fluid surrounding the cells to always be hypotonic, meaning water will always move into the cell. Once again, This is a passive process and no energy is required. So this is osmosis in plants, especially in the roots. And lastly is the active transport. Active transport is the movement of dissolved solutes across a membrane against a concentration gradient, which is moving from low to high concentration against a concentration gradient, or the movement of larger substances across a semi-permeable membrane. This process requires a carrier protein and energy in the form of ATP, and carrier proteins in the cell membrane are required. ATP is what we know of as energy. It comes from cellular respiration. Cells that undergo a lot of active transport will therefore be packed full of mitochondria, the organelles within which respiration occurs. The fact that active transport allows cells to move substances into them against a concentration gradient means they can absorb substances such as ions and sugars from very dilute solution. So this is an illustration of how active transport does. So why these processes are important for plants? In most land plants, oxygen and carbon dioxide enter through the leaves while water and mineral salts enter through the root system. Substances can move into the root in several ways, with the principal method being osmosis. During osmosis, free water molecules pass from the soil into the epidermal cells using the root hair membrane. Looking at this process at the molecular level, we see that the smaller water molecules pass easily through the selective permeable membrane of the epidermal cells, which I will show you in the next slide. Diffusion of mineral salts occurs at the same time. Facilitated diffusion occurs when important molecules pass through the membranes via special holes called channels. Active transport of other molecules may also take place in the root hair. Depending on the needs of the plant, active transport requires energy, as the required molecules must be pumped across the membrane against their concentration gradient. So these are the pictures that show how or why these processes are very important for plants. Thank you for listening. So this is the end and hope you like it. And goodbye, sir.